Welcome to the theEWpodcast. I hope you're staying safe and healthy out there. Today's episode is somewhat relevant to the coronavirus, but since it was recorded in early February, the virus is not explicitly mentioned. But maybe for some of you, that will be a nice break from the virus. We're seeming to get plenty of it in our daily lives anyway. Today I speak with my good buddy, Kevin Hackett. Kevin works in the world of genomics and has a degree from Clemson University in biochemistry and genetics. In this episode, we talk about gene editing and CRISPR. This basically turned into a gene editing 101 course. We start very basic with what genetics are, and we touch on topics later in the conversation, including how CRISPR works, what types of diseases it might be used to cure, and what the future of gene editing looks like. If you're interested at all in this topic, but like me, have very limited knowledge in it, then this podcast should be right up your alley. In the show notes for the episode, I've also provided some links to resources online regarding some of the things that we talk about in the show, including the Chinese researcher and the SMN1 gene. So you can visit theEWpodcast.com. That's theEWpodcast.com. Find the episode with this show, and there will be the links. Also, if you're listening on iTunes or Spotify, you can find the links in the description. All right, one piece of housekeeping. I have started an email newsletter, the first iteration of which will be sent out this Friday. This will be a monthly correspondence. It's going to be similar to Tim Ferriss's 5-Bullet Friday, if anyone is familiar with that, except less frequent. In the newsletter, I'll provide links to the latest EW podcast episodes and blogs, if there are any. And in it, I'll also provide some links to various things I'm reading, listening to, or doing, all in the vein of mental health and or the mind. You can sign up for the EW Podcast newsletter at theewpodcast.com/newsletter. Without further ado, here is my conversation with Kevin Hackett. Cool. Thanks for joining me, Kevin.
Thanks for having me, Eric.
A little sip of coffee there.
Yeah, let's get started.
Yeah. So to preface this whole conversation, start it off, I just want to kind of express why I'm interested in what we're going to talk about. And we're going to talk about gene editing and CRISPR and all that good stuff. Mm-hmm. So the reason I'm interested in it is because I just find disruptions to society very interesting, mm-hmm. like things like AI, mm-hmm. The green movement in terms of climate change, like all these things where we're going to have massive shifts in the way people live, mm-hmm. really interest me. And I think genome editing specifically interests me because it's not just a technological advancement, it's like a species advancement. Mm-hmm. You know, we're changing our species. So I think that is just fascinating, and that's kind of why I'm interested.
Excellent. Yeah, I'm happy to provide as much context, background information, as well as just kind of firsthand knowledge as it relates to genetics and specifically gene editing. I feel as though I have maybe a little extra knowledge in comparison to kind of the normal landscape of people, simply because of my background. I have my degree in genetics and biochemistry from Clemson University. I work for a local biotechnology company that is in the world of genomics and genetics, not necessarily specifically gene editing, but we do provide technology that may be employed by people within the world of genomics, whether it's for gene editing or simply genetic mapping. And I am very interested and very passionate about this topic. I mean, since essentially high school, when I learned about the complexities of the genome, and that really we were at, and still are, in my opinion, at the very beginning stages of what will be a massive kind of opening of knowledge and access to genetic insights and overall scientific research that could benefit the human condition, benefit the human race, benefit a number of species across the world, and both in the realms of therapeutics and clinical kind of application, as well as agriculture, and just essentially optimizing who we are as a species, potentially, if done in the right way, as well as kind of what we get out of the place in which we live, the planet Earth.
Nice.
Yeah.
Thanks.
Yeah.
Thanks for sharing that. That's
sure.
awesome. So to get us started, I have this icebreaker.
All right, roll.
I
Let's
was
go.
given a deck of icebreakers, and I found this one prepping for our conversation. I thought it was a good way to start. If given the choice, would you live forever? Common question, but I think it sets us up nicely.
I don't think I would. I mean, I think that it's a very big question, and a lot of it comes with, I would say, maybe it depends. A lot of it depends. What would be my physical condition throughout the course of forever? But imagining living forever, it's kind of a difficult thing for me to swallow because, we are on. I think the finiteness of life empowers us to live it, boldly. It can, and to get the most out of it, and maybe living forever. Yeah, maybe there are certain things that you don't want to see, that it's kind of your purpose to be on Earth for a certain period of time to get the most out of it. And maybe our brains where we're at right now and what we're able to comprehend, just like maybe it's not meant to be forever,
Yeah.
I think the finiteness of life, as I mentioned, just it should embolden you to get the most out of it, and not just say, I got another couple thousand years. I don't need to go for that, I don't need to go smell the flowers today or whatever, stop and smell the flowers sort of thing. So, I don't know if I'd go forever. I kind of like what we got going on right now. Maybe we can extend it, but if we extend life in some shape, form, or fashion, then the quality of life also has to be there too, right?
Yeah.
How about you?
I agree with you there. I'm like back and forth on it. It also depends on loved ones as well. Are they going to live forever? Is this a friendship-wide thing, or am I the only one doing it? That would definitely influence my decision. But I lean no, just because, like you said, the finiteness of everything is like important to actually enjoying it, I think, and to appreciating.
Right. And who knows, what our condition will be, or the condition of the Earth for that matter, I don't want to say I'd live forever if something happens and I'm the only one here.
Yeah, for sure. Not a future I want to be a part of.
Yeah, kind of scary.
All right, so let's just jump into it.
Cool, let's go for it.
So this can be kind of a dense topic, and I am a journalism major. I have no scientific background. I'm just, like I said, interested in the topic. So I want to make this just as easy as possible to understand. So let's just kick this off with the basics here. What's the genetics? What are genes?
Sure.
Let's lay some groundwork.
Okay, great. So genes are essentially encoded information into the nucleus of the human cell. We're gonna use we'll use humans as kind of our case study or our model organism of sorts. So genes are the information that is essentially, a lot of people use the term blueprint for life. Within the nucleus, or the center of the cell, you're going to find that deoxyribonucleic acid, or DNA, is encased across a number of chromosomes. So genes are essentially the singular take on a set of genetic information, meaning A's, T's, C's, and G's, or base pairs, are orchestrated and put in a certain order that when read or when accessed by different proteins, as well as RNAs or ribonucleic acids, they can be translated into physical form, essentially, or something that provides a function in the body. So genes can be anywhere between, let's say, 100 base pairs, ATs, with C's and G's, up to thousands. And sometimes it is the coordinated expression of multiple genes that then turn into a specific protein. It's not just maybe one gene equals one result, blue eyes.
Right.
Sometimes it is a multitude of genes acting in kind of an orchestra together in order to create what is called a phenotype. A phenotype is a physical expression or culmination of genes. A genotype is essentially it's the characterization of the type of gene that you have. So let's contextualize this again in the form of a human. So a human has 23 chromosomes.
Every human has 23.
Every human has 23, and that's what gave name to the 23andMe. You also have your X and Y, or your sex chromosomes. Males are XY, while women are YY. So, I'm sorry, XX. So in those 23 chromosomes, there are multiple loci or locations. That's what we refer to them. Essentially, it's like a map of the genes. Now, within those loci are specific genes of interest. So you start big, you got your chromosomes, you drill down, you've got your loci or locations, and then within those locations, you have the specific genes. Now, we have just started really mapping the human genome. It was successfully done a little over 10 years ago. I think it was to the tune of $13 million, and now we've gotten it down to essentially $1,000 a genome. That's the rate of advancement as an acceleration of technology has allowed us to reduce the cost per genome dramatically, which has enabled so much more groundbreaking research. And it not only in the form of, say, gene editing, but just identifying what is where and what does it do. And once we understand that, then we can, kind of move down the course of research, or further the course of research to say, if we do this to this gene, then what happens? And that is essentially the birth of gene editing. So there are, like I said, 23 chromosomes. There's around 20 to 25,000 genes across those, across that genome. Go ahead.
Does every human, we have the same amount of genes in each chromosome?
Really, really good question. We do. We have the same amount of genetic material as I would say. The large majority of humans have the same amount of genetic material. I would say the variability there is, say, with genetic mutations like trisomy 21, which is 3 of the 21st chromosome, that's what gives way to, say, something like Down syndrome. So there are mutations and conditions that can result in more or less genetic material, but that typically is associated with a disease or a manifestation, a phenotype of some sort that can be deleterious or, perhaps have additive effect to the condition of the organism. A good example of that, so we as humans, we have the same amount of genetic material, but then there are certain other organisms, like, say, the oyster, that can have a diploid genome. So that's essentially, again, 2 sets of parents come together. So haploid come together to form a diploid, meaning you get half of your genetic material from your mom, half your genetic material from your dad. You put it together, you're a diploid organism. So there's essentially 2 genomes are coming together to form your personal genome. Something like the oyster, they've found that it has a triploid genome, or can have a triploid genome. So much more genetic material. But that said, I think what's important to note is that genetic material, or amount of genetic material, doesn't equate to sophistication or complexity necessarily. It is the expression of the genome that truly gives rise to highly complex organisms and really differentiates us from each other, as well as us from, say, the ape. We have such, I forget what the actual figure is, but the similarity in genomes between us and the chimpanzee is it's astounding. But it isn't necessarily that we have all of such similar DNA. It's that the parts of that genetic material are expressed at varying levels to differentiate Eric from Kevin, Kevin from Chimp.
Right.
Go ahead.
So it's the combination of the A, T, C, and G is what is giving us the differences. It's not the quantity of genes. It's how those letters are paired up within each gene.
Precisely.
Okay.
So like I said, you have 23 chromosomes, you have your sex chromosomes. Now those are going to be expressed in varying patterns and varying times under varying conditions. Certain parts will be accessed and expressed to, say, create more immune cells, or to proliferate more growth within a specific area of the body, or for that matter, when something goes wrong, like a cancer cell, maybe certain genes are being expressed in rapid succession, so much so that the body can't suppress that or stop that, and that's what gives way to cancer or a tumor.
So they're not always on.
Certain genes are not always on. Certain genes are kind of constantly running, and certain genes are expressed in only certain conditions, certain points in our lives, under certain duress and circumstance.
So why? Well, I guess let's start with the why. Okay. Why would someone want to go in and change their genetics?
Yeah, that's a good question. I think that the most obvious is to improve maybe something that they were born with that impairs their life, or again impairs the human condition, impairs their health. Good examples of this are going to be, something like blindness, or some sort of heme condition, like a blood disorder or blood disease, or for that matter, cancer. We do want to possibly go in and specifically change the genetics of a tumor in your body so that it can be perhaps more accessible by the immune system to fight it off naturally. Now, I know that a lot of people think of gene editing, where a lot of people quickly go with that is, selectivism, and being able to say, I want, blue eyes over green eyes.
Or tall or short.
Yeah. And I think that the kind of the designer baby take on gene editing often gets popularized in headlines because of what could, clearly be considered kind of like a, selective advancement of our species, and for almost aesthetic purposes, as opposed to those that can bring a lot of added value to people's lives, and allow them to enjoy their lives and like the finite time that we have here on Earth, unless we do you pull that card again and we decide to live forever.
Yeah. Who knows what will change?
Yeah. I think what may be helpful is perhaps an example of what gene editing would be.
Yeah, let's
sure.
do it.
So gene editing can come from essentially 3 different flavors of it. There's going to be gene editing where you're replacing a certain gene, or repairing, let's say, a gene that is off, or where you're supposed to have an A, you have a G. And because you have that, let's say the retinal cells, it's the cells of the retina are not as sensitive as they need to be, causing blindness, causing maybe misformation of the rods and cones in your eyes. And the idea here is maybe we can introduce a changing of just that one G to an A, so that those rods and cones are actually, when they're essentially replaced or turned over by the body, and we do have, let's say, I think 20 to 40,000 cells turning over in our body at regular succession. Our body's constantly repairing itself. So if we can somehow teach the body how it should be repairing itself, then we can repair those rods and cones in your eye. Another consideration of gene editing is going to be a knockout, meaning go in, there is maybe an extra couple pairs of a certain gene. Maybe a gene has been repeated. You got 2 sets of it, which results in you producing twice the amount of retinal cells. We'll stick with that. And now you're hypersensitive to light. So you can't look at light because you've got too many of whatever cell in your eye. So perhaps now the approach is we need to remove one of those sets of genes so that you have the proper amount of retinal cells rather than too much.
Is that taking away genetic material though? Is that because that was something I was getting hung up whenever I was looking into this, was that the knocking out of genes, because doesn't that leave you with less genes then, or is it being replaced by something else? You would, at a very micro scale, be reducing,
a gene, but think about it this way. You have 4.6 billion bases across your 23 chromosomes. One less base, or maybe a couple, in order to have the body work properly. Technically, yes, you are going to have less genetic material because you're telling the body, I don't need this one adenine here or this one, So on a micro scale, I guess, yes, you'd say you'd have less genetic material, but in total mass, I don't. That one less base probably is going to have. It's going to get you back to normal rather than it would, being kind of feeling left empty-handed.
Yeah. I'm just like imagining cells with less genes just like liquefying and disappearing. Not having enough internal structure.
Yeah, totally. No, I get what you're saying, and the body is incredible in the sense that the repair mechanisms, the the pre-programmed means of cellular death that we have. If something is wrong in a cell, the body is so immensely incredible in terms of its capability to identify wrong cells and remove them, or for that matter, to repair them internally. It also creates a good example of how gene editing happens naturally in the body through natural endonucleases and enzymes. The research that we've done has taught us essentially what to go after in the future and how we can, for lack of a better term, hijack naturally occurring gene editing mechanisms in the body to do essentially what we're asking, doing our bidding. The CRISPR technology that is really getting a lot of buzz right now is an incredible technology, but was actually hijacked from a naturally occurring virus where an endonuclease, or it's essentially an enzyme that works within the nucleus, it is given essentially a target strand of DNA. We were able to say, okay, this one gene, let's say it's 100 base pairs, and on the 54th base pair, we notice in your body that, or in your eye, that you have a C instead of an A. Well, we're going to give you that. Actually, we're going to attach a strand of the accurate interpretation of that DNA strand of that 100 base pairs. They're able to essentially give a transcript to that endonuclease. And endonuclease then has essentially a map of what it's looking for in the cell. And that's why CRISPR is so groundbreaking, is because it's so specific. It can find that very specific 100 base pairs. Again, this is just a pure example, and I'm not being overly scientific here, but it can find that very specific area in your genome that it is intended to go in, bind to, cut, and repair, essentially turn into the correct version of that transcript. So, now, with that said, we have, like I said, this level of repair happening constantly throughout our body. With so many cells being constantly turned over and created, we would, to your point, dissolve and vaporize had we not have some sort of naturally occurring repair team at work within the nucleuses of our body and throughout the entirety of our body at all times. We've essentially just been able to say, hey, this one works really well. What if we teach it where to go and give it the information necessary so that it can find the specific mutation that we feel as though is worthy of repair?
It's insane.
Yeah, it's pretty wild.
It reminds me of like a police dog or something that you give him the scent of what he's looking for, and then he just goes and gets it.
That's exactly it, except the dog goes in and
goes and does the work. Makes the arrest and everything.
Exactly.
Takes him out. Yeah. So you mentioned the destroy was one, and then there's also the replacing. And
then there's the new gene, essentially introducing a new gene that was not there. Now, that may be because 99% of the population is born with that gene, but you were not. So what we want to do is essentially provide a means by which we can introduce that gene into your genome so that now you're whole and you're not less a gene. Those are the three kind of flavors. You got your replacing or repair, your knockout, and then your introduction of new genes. Some applications of this, like I said, can be in the cancer world. They can be in the blood disease world. I think they can be in the rare disease world. There was, I think, one of the most noteworthy or headline-worthy applications was the Chinese scientists who, using embryonic cells, went after the CCR5 gene. The CCR5 gene is actually, it is a gene that is naturally occurring within a very small sliver of the human population that provides essentially a natural defense from HIV. It is CCR5 is a surface binding protein. It has this innate immunity to the HIV virus. So what this doctor did was essentially using embryonic cells in a controlled environment and went in to see if he could introduce this gene into an embryo that previously did not have that gene. So really wild stuff that they're able to get to that point and now introduce into an embryo a genetic material that was accepted by the rest of the genome at such a nascent stage of the embryo, essentially, so that it could you're affecting the essentially germline cells. These are the cells that turn into everything else in your body, right? So that every cell in your body now has that added gene.
Would offspring of that person have those mutations as well?
Yeah, so really good question. There are two different kind of states of cells in the body. Well, there's a million states of cells, I'll take that back. But you have germline and you have somatic. So somatic cells are going to be the ones that are essentially only tissue. You can't pass them on. It's like me or developing a therapy for, let's say, skin cancer on your arm. You're not going to pass that skin cancer down to your children. And if there's a therapy for that somatic cell and editing the genes of those somatic cells, that isn't then going to be absorbed by the rest of your body and then into your sperm and then passed on to your child. But contrast to that is your germline cells. These are the cells that essentially make everything up within you and that you pass on to your offspring. I believe that's such a kind of budding part of a human, essentially you start with one cell. If you can edit that one cell, your very first cell, it's then going to proliferate and divide into all of your cells. A lot of the therapies that they've developed right now are focusing on somatic. I think that somatic is a much safer place to start, right? Fixing, say, esophageal cancer or AIDS. And in order to, I would say, progress through this technology, explore this technology and its application safely, we should start with something that is finite and not going to be passed on generation to generation. Let's start slow and learn as we go. And then, perhaps they will get to a point where they find let's say, if there is a gene that you know that you are going to pass on to your offspring, and it would be, it's essentially going to ruin them. It's not going to give them the life that you wish to provide them, and it's not going to it's going to slight them. Where the ethical kind of quandaries come to play is like, well, how bad does it have to be before we actually feel as though we have the level of confidence where we can go in and edit this thing and feel good about it and know that we haven't crossed a boundary and played God to a certain extent. What I think I know about the Chinese researcher is that it was done in such an environment that the cells weren't ever intended to be grown into a human fetus. It was in a very controlled embryonic stage, very early stage, just to see if the uptake of the genetic material would actually stick.
What was the reason for all the uproar around that? Was that because of his secrecy in which he did the experiment, or was it because it was an embryonic change?
I think it wasn't really in secret too much.
Okay.
And I think it was the fact that he was the first to actually go after a human embryo. And you were essentially altering the cells, and now this person you're changing their deck of cards that they have to live with for the rest of their lives. As opposed to maybe changing the seat that they're at the poker table, that is somatic versus germline, how, and changing a part of you that are a tissue versus something that affects every tissue in your body.
Does CRISPR ever miss? Does it ever?
Good question.
Edit the wrong part?
Yeah. Really good question. I'm glad you brought that up. So yes, that is one of the long-term concerns, is the fact that we haven't seen the long-term data and the long-term effects associated with gene editing or CRISPR. Again, it's such a new technology that we don't really know what could happen, with anyone who is given this therapy over not just in a short amount of time, oh, you're cured of your XYZ disease. Well, did that change actually propagate a new disease later in life? So one of the concerns is off-target edits, and that can be for a number of reasons, but typically that transcript or the kind of the scent that you gave the endonuclease or CRISPR, the CRISPR enzyme, sometimes that can be extremely similar to other parts of the genome. Sometimes a version of that gene can be represented in multiple places throughout the genome. So off-target edits are yes, a very big area of concern and something that should be considered and explored quite a bit prior to launching therapy. And that's essentially why we're in this kind of golden era of discovery and really understanding what this type of gene editing therapy can do for us. And what we do there is typically we'll sequence a genome or sequence a specific gene, because you can do genetic mapping or DNA analysis, genetic sequencing. You can do it on a large scale, and you can sequence your entire genome, or you can pick a very specific area of the genome to target to sequence. So that's obviously a lot more cost efficient, right? 100 base pairs versus the 4.6 billion base pairs that you have. So what we can do is we can sequence it before. We can essentially go in, okay, we know this is your condition. We're going to confirm this by sequencing the 12th chromosome, this area of the 12th chromosome, and then provide the therapy. Let's say it's for your eyes. And then they can do a second follow-up sequencing run to essentially confirm the change in that base
Checking
pair.
your homework.
Exactly. And again, the rate of advancement within the world of genetic sequencing has both in terms of the cost going down, as well as the specificity of confirmation in genetic sequencing has allowed us to not just throw these technologies in the body like CRISPR, but to do it in a way where we can do it iteratively and, small bit by small bit. And also to, let's say, back to kind of the off targets, let's say that 100 base pairs that you intended to change. Well, let's confirm that, but then let's also sequence the rest of your genome to see what else changed as a result of the introduction of something, say, like a CRISPR-Cas9.
Is it more helpful then to have more base pairs in the area you're trying to change because it diminishes the opportunity for there to be some overlap? You know what I'm saying?
Yeah.
So the RNA you give to the Cas9 protein maybe is a little bit longer so that
more specific. Yeah, it would. Specificity and the ability to accurately target and lock onto an area of the genome was going to be enhanced by having more of a transcript or more of a scent to go after. Now, with that said, longer RNA molecules are what would be connected to the CRISPR-Cas9. The longer they get, the more fragile, the more they are susceptible to, let's say, naturally occurring other enzymes in the body to say, okay, well, what is this? What is this endonuclease? Actually, I don't recognize this as something that's normally happening in the body. I'm going to now eat this thing or get rid of it. But longer oligonucleotides, which are essentially a long chain of nucleotides, your scent, the longer they are, the more essentially area they have to be broken down, or for that matter, RNA itself can misform. Perhaps now we've introduced such a long strand of DNA or RNA to the CRISPR-Cas9, maybe, and we think it's going to be so specific that because it has this very long strand, but halfway through kind of targeting the area of the genome that we want to edit, maybe another enzyme finds it and cuts it in half. Now it's half as specific. So I know that, again, the rate of research is very impressive, but they're trying to find what that exact amount. Exactly.
So is there a limit to how few base pairs can be used for a swap and replace?
It's a good question. Offhand, I don't have it in my head. Although there are 100 base pair genes. There are a couple hundred base pair genes. There are certain genes that work, again, kind of polygenically, so multiple places throughout the genome. That come together, multiple genes come together to form one phenotype. I also know that you don't want it to be too long so that it potentially could overlap with another gene, or for that matter, if it doesn't, you want to find just the right amount of fit and not requiring the potentially such perfect fit that it's always going to miss. If there is, say, another gene that overlaps or is right next to it that does have a small mutation, but perhaps that mutation isn't as deleterious or can negatively impact the body as much as the one that you're actually going after. So increased specificity may actually lead to a decrease in overall attachment and correction.
So just kind of in review to paint this whole picture using the dog metaphor.
Sure.
So you have, we'll say the bad guy is your DNA that you want changed.
Okay.
So you're giving the dog your enzyme, your CRISPR-Cas9 dog enzyme, a copy of the DNA, which needs to be replaced.
Right. The DNA of interest. Yeah. Yeah.
And so that then the CRISPR-Cas9 is put into the subject's body, and it sniffs out that scent within the nucleus, the DNA, and it either changes it or
knocks it out. Yeah. Yeah.
Okay. And so then from there, how long until the changes are seen in the body? How long does it take for that to be expressed?
I from what I've heard, it's pretty miraculous in terms of the turnaround. I don't want to say like, you put it in 10 minutes later, you're healed. But I did look up one example of the SMN1, it's a motor neuron disease that leads to spinal muscular atrophy in essentially children. It's a fatal disease with this one gene, SMN1, and it affects the muscular tissue and the connective tissue for various organs. And ultimately what leads to patient death is the lungs don't have the essentially the muscular capability to contract and to contract and deflate, I guess. And so the child essentially suffocates to death internally. But what they found in some of these patients, and they're I think as another caveat or as a caveat, I think it's important to note that this technology is being implemented in very limited kind of case studies, but for the people who don't have any other option, and that's the place we should start, is essentially giving someone a chance. But in that circumstance, they found that children are breathing and upright weeks, months after this initial therapy. It's really, and it has just blown physicians and researchers away alike.
So what is happening in that waiting period? That's just the cell dividing itself and creating. Once that new DNA has been created in enough cells, then it becomes expressed. It becomes the dominant.
Exactly. It becomes it becomes the one that is essentially the new norm.
Okay.
Yeah.
We've mentioned before some of the diseases that are being thought of. You've mentioned
heme diseases, things that affect the blood.
Is there any in particular that's being more talked about than others? Any application that has the community more gung-ho than the rest?
Sure. I think that in the world of immuno-oncology, which is using your body's natural immune system to attack cancer cells rather than, say, bombarding with radiation or chemotherapy, is where a lot of people are excited, and they're seeing a lot of potential applications for CRISPR. So there is a protein on the surface of tumors. Essentially, PD-L1. PD-L1 gives way to this surface protein, and what it does is it's a pretty mischievous little gene in a tumor cell, in the sense of it tells the immune system, no, don't attack me. It enables the tumor to essentially be masked from the natural immune system. So the idea here is, well, can CRISPR be leveraged to knock out that gene so that now the body's natural immune system recognizes this tumor that's maybe been there and growing, but the entire time it's telling the body, no, I'm natural, I should be here, I'm just growing at a rapid pace. And by knocking out that gene, we can actually give the body's natural immune system the visibility and capability to now attack that tumor cell naturally. Kind of unnatural that we're introducing CRISPR, but natural in the sense of we're tasking our own immune system to repair the body.
And it's using like you said before, something that's already found in nature.
Right.
The protein is a naturally occurring.
Right. Right.
So it's all really natural, but it's kind of just being instigated by us.
Right. Right. And I think that's really where I think a lot of innovation throughout all of the medical community, that's all it is really over the past 100 years. It's just identifying naturally occurring things that happen in the body, things that benefit us, and then being able to harness that capability for the benefit of other humans who maybe don't have that capability, that protein, that enzyme. Other areas of application are going to be around rare diseases, blindness, cystic fibrosis, and as you mentioned, HIV and AIDS. I think that those are kind of the biggest buckets of application that people are most excited about.
So is the act of changing traits, genetic traits, is that something that's, I mean, in like physical characteristics, is that something that's not even being kicked around in the scientific community? Is that more a sci-fi headline?
I kind of perceive it as a sci-fi headline, but at the same time, physical traits can be manifested in any number of ways, and I think physical traits at the level of like almost aesthetic interpretation, eye color, hair color, muscular build, things of that nature. I think, at this stage, of course, it's in conversation because it's like, well, this technology is obviously going to lead to, at some stage, the ability to be selective in terms of what we are or maybe what we aren't. But right now, the biggest area of focus is the things that are hurting people the most. And I think that where is also having the biggest impact is in patients who don't have any other options. This is something that they can do as a last resort, or for that matter, maybe it's going to be a first line therapy in the future. But how do we get there? We start with the people who do not have access to any other therapies. And again, they are at their last resort. And we are validating technology and validating the implementation of CRISPR-Cas9, understanding what side effects are out there, understanding the efficacy, understanding the safety of it. And then once we learn quite a bit through these kind of very, small clinical trials, small patient populations, then we will start to, I would imagine, start to introduce other considerations of CRISPR-Cas9 and just gene editing as a whole.
Is the main obstacle then just figuring out the safety of it? Is that kind of the main obstacle that CRISPR faces in becoming a public or more widely used technology? Yeah,
I think it's, well, one, it's safety, but then it's two, it's clinical validity, right? How much impact can it truly have? And based on that impact, what is the downside? What then are the safety concerns? Is it better than what is other options that are out there right now? That's what the FDA goes through. That's kind of their process is, okay, we know it's a new therapy, but does it do it better? Does it do it safer? Does it provide patient benefit? Does it provide some sort of unique differentiating factor in comparison to what's currently in the market right now? Or is it, just so similar to another therapy that it's not worth putting this through because the other therapy that is out there is proven and it's proven to be safe, proven to be effective. But yeah, I think right now it's a balance of efficacy and safety.
Is our genome close enough to something like a mouse for animal models to be used in testing certain human applications?
Yeah. At the most basic level, yes, we're all A, T, C's, and G's. We're all carbon-based life forms with a double-stranded DNA helix that comprises a number of genes that turns into a number of chromosomes. But a mouse has a different number of chromosomes than us, different sizes, and even plant species have far bigger genomes than we do. But at its most basic level, the actual function of the technology is essentially going in and doing the same thing that it would do in a mouse as it would to us. But because of just, the different functions of the body in a mouse and the human, because they're so vastly different, that's why we do these controlled trials within, say, a model organism like a mouse. Then you go to a smaller, kind of cohort of patients and kind of the first patient trial or human patient trial, and then you scale up from there. But it wouldn't be as simple as saying, the CRISPR-Cas9 therapy that we provided a mouse to cure of diabetes is going to be the same one that we give to a human to cure their diabetes. Right. It's going to target a different part of the genome, but at the same time, it's targeting measures and the actual mechanism by which it's going about that change is essentially the same.
So they can use some of that information in human models.
Absolutely. You got to start somewhere, and then you grow from there.
How much longer do you think it'll be before we see this being used more widespread?
Yeah, it's a really good question. I think in the next 10 to 15 years, I think it will be not a regularly accessible therapeutic option, but something that will be available to a specific set of patients and based on essentially whatever their condition is. So I think it's going to become more of a readily acceptable therapy and proven therapy and prescribed therapy in the next 10 years for sure. I mean, they actually just did the first FDA-approved human trial with CRISPR recently, and that was August of 2019 is when they did that, and that was that SMN1.
Was that the lung?
Yeah. The motor neuron disease causing spinal muscular atrophy.
Oh, okay.
Yeah. So I think it's going to become more and more prevalent. Now, the interesting thing is, where will we see it? Where will we see the prevalence grow? Likely not in the U.S. Yeah, there are regulatory bodies or such that, and for good reason, are they want to be a part of that process. They are going to ensure that the research and the therapy are done in controlled environments so that we can safely understand the true effect of this technology. In other areas of the world, like your China's and even in Europe, the regulatory bodies are, I would say, are not as conservative. They adopt. They're early adopters of new technology much more so than the U.S. And this varies from country to country. It's not the U.S. is the harshest regulatory body, like with the FDA and whatnot. Yeah. But I would say that there's a lot more discovery happening via earlier adoption and through more trials outside of the U.S. than, say, in the U.S.
To what extent is the global scientific community working together on this, or is it a country?
That's a good question, and I think that there is a lot of information sharing across the globe happening, and there's a lot of partnership on this technology because of the promise that it holds. I'm unaware of, say, like a CRISPR team, a unified team across the globe, and a lot of researchers are obviously going to be protective of their discoveries so that they ensure that the data and the understanding and the discovery is used in the right way, but also because of potential commercial application and being able to gain the fruits of their labor. But I do see a number of researchers around the world coming together for a number of other technologies or therapies or cures. But this is like really we don't even know what people are trying to discover is, okay, how we know what we want to cure, but how will this potential therapy improve our ability to cure that, again, safely, efficacy and safety, and the sharing and that information. I mean, daily data is coming out, and the more you publish, the more notoriety you get, and then the more funding you usually get. So it is in the scientific community's best interest to publish your data as quickly as possible. Yeah.
This just seems to me like something governments around the world would be so invested in I don't know, kind of a darker view of the world, I guess, makes me feel like it becomes like some sort of arms race or something. I know I'd sent that in the notes. Is this like the next arms race where it's like you're trying to make your populations genetically superior? You got to kind of get into that eugenics field, which is a little scary. Is that something that frightens you or your coworkers or anyone you know at all? Is that like the worst case scenario?
Yeah, obviously we want to make sure that this technology imparts positive value to the human race and to the world. In terms of widespread adoption to the point of eugenics, I would think that we, as a human race and as kind of watchers of the world, would be aware and involved in discussions and actions at a global level to inhibit and to control that. But when you extrapolate examples like this, of course, you're going to end up at some potentially far out place. So I think it's natural for the imagination to go there. But I don't see the technology moving so fast that we can't keep up with it and be able to essentially control or throttle its application to the point where it would get there. We'd be able to hold it and guide it in a manner where it is not turning humans into a super race, Yeah. But there's so much subjectivity to that, right? Like, what is a super race? If I'm able to give you the CCR5 gene CRISPR-Cas9 editing and now you're immune to HIV, we're one step closer to a super race. So I can see it from both sides. It really is it really is an interpretation of what someone feels as though a superhuman or optimized human is. Is it just being able to live your life in confidence, knowing that you're less likely to get a disease, or to be a superhuman? I could say you have to have increased muscle mass and like be beautiful, and you're not susceptible to diseases. So I can see the far ends of the spectrum, right? One simple change, CCR5. Optimized super soldier, and both of them are genetically edited, but both of them can be considered superhuman, or at least better than the normal human condition. And I think the other kind of long-term implications to think about are not just, well, what if I do reduce the susceptibility of HIV globally? Awesome, great, we did it. We knocked it out. Now the human lifespan has gone from 72 years to 75. Do we now have the resources to be able to sustain that extra 3 years per person on the planet? So, or, do we have enough, because now you're going to live to 75, now you're going to increase the human population and just density and the number of humans by 2%, because just people are living longer, people are having more kids, and so now do we have, enough kind of, like I said, natural resources to sustain us, or actually can we optimize to a point where we are putting ourselves out of the game? Where we've now gone so far in pursuit of good that we actually run the risk of net negative impacts globally, because of habitable space to live, water on Earth, food to eat, air to breathe. With these small changes, what does it do at a macro level? We're talking very, very, very micro, even at the genetic level, and then micro in terms of the single patient level. What about the global population? What is this small tweak in human survivability, susceptibility to disease, the ability to see light better? If we change that, what does that really do long term, not just for that single human, but for humans at a grander scale?
I guess we'll find out.
Yeah, I guess we will.
Has working on this and coming to understand it changed your perspective on life? And if so, in what ways?
Yeah, it has changed my life in a way that has challenged me and forced me to think about not just, as we mentioned a few seconds ago, singular impact, but really bigger impact. What can this do for the human race? What can this do? What will this do to planet Earth? What will the adoption of a technology like this, be it fast or slow, affect the way new technologies are adopted, both in a regulatory capacity and a research capacity in the future? this technology has potentially groundbreaking status, right? But we have to do it. We have to adopt it in a manner that is cautious and weighs risks, and not to be so wrapped up in it that we're kind of blinded by the positive upswing and what it can bring. So it's made me think about things a lot more long term. It's also made me realize that regulation will come. And I think it's important for us to not turn to our research community and say, whoa, everyone stop. We need to talk about this. Like, everyone stop what they're doing right now. Put your pipettes down, because we need to put some laws in place.
Your pipettes.
Yeah. To halt research in order to discuss every single situation, circumstance, or scenario, and decide what's right and what's wrong, to halt all the research just to have that conversation, I think is pretty short-sighted. We need to empower the research community to continue to push on all fronts so that we can explore every scenario. But we don't really know what scenarios are going to come down the pipes if we don't continue to push research and consider the applications of the research and technology. For instance, we spoke about HIV, but of course, as we begin to push on research, this bioethical kind of considerations come into play where HIV, yeah, that's great. Let's attack that. But when you extrapolate, the potential applications, you will ultimately get to that blue eye. I want to be taller. I want to be more muscular. I think if we give someone a deck of cards that is devoid of any life-threatening condition and allows them to live a life worth living, that is where we need to go with this. And if we start to edit the deck so that someone can be, say, smarter, well, again, what is the long-term risk of that? Not necessarily what are the, what extra neurons or, neural networks do they get, but I mean, you could take it as far as, well, let's say they become a brilliant, a brilliant scientist, and they become so wrapped up in their research that they get some form of psychosis or something. Essentially, you don't know. Even if someone is able to be more intelligent, well, where does that intelligence bring them? Maybe it brings them to the loony bin because they're so smart they drive themselves crazy. I think it's important to give our future generations. Well, one, we should leave the earth better than we found it for them. Secondly, we should enable them to live a long, prosperous, within means long, prosperous, and enjoyable life, but not do it in a manner where we are orchestrating their lives for them. We're predestining them to be something or someone, or for that matter, expecting them to be something. Because if we give someone enhanced intelligence, well, then doesn't that also kind of set some high level or some very high expectations of that person? If they don't live up to those, then, what does that do for a kid's, just overall, demeanor and psychological state, we upped your intelligence, man. You should be killing it in this math quiz, no, that's no. And another thing too is there is our genome and then there's our epigenome. So we really go all the way back to the beginning of our conversation. Yeah. So the epigenetics it's essentially the over genetics. So we have our entire genome. Let's use a book as an analogy. So you have your entire genome from, we said 4.6 billion bases. So first letter of the book, base 1. Last letter, 4.6. So throughout the course of your life, you are not reading every single letter at every single moment. Certain chapters are accessed, certain parts are read. And the epigenome is essentially the overgenome. It is the lens by which you look through to look at that book. And it is the variable expression of the genome and the why behind it. So why do we have all these genes? Why do we have, and not every part of the genome is essentially an important coding region. Some we don't even know what it is. There's 1,000 base pairs here, and they do nothing at all, and they seem like nonsense. We don't fully understand it, but we know that for very specific reasons, certain parts of the genome are accessed or hidden at certain parts throughout the life. So it's kind of this over genome that exists, and it's not just the book that you were given as a kid, when you came out, and here's your genome, here's your book. It's essentially, the epigenome is how you read that book throughout the course of your life, how your body reads that book and accesses that book and expresses that book. So it's not always as simple as one-to-one. Back to the intelligence gene consideration. If we give someone, let's say, 5 copies of the intelligence gene, maybe the body actually can't interpret those 5. It doesn't have the other genes necessary to create the proteins to access and to express those other 5 intelligence genes. So it's not always one-to-one. And that can also lead us down the conversation of nature and nurture. You're born with a certain set of genes and a certain biological blueprint, but through the course of your upbringing and through the environmental stresses, through what you take into your body, certain parts are going to be accessed, or for that matter, not accessed. So again, it's not always one-to-one. And right now, we're really helping define how close can we get one-to-one, especially with the consideration of therapies like CRISPR.